Hi, everyone. So this is the weekly announcement for weeks 2 and 3. Week 2 would have been everything that was last week all the way up through last night at 11:59. And then I will cover what's due this week because you do have a lecture exam that's due June the 10th. So what I'm looking at is the chapter 3 homework assignment results. And if you notice, it has the score for each question over here. So you see this question here that says chapter 3 multiple choice question 10 had an average score of 37.5%. And then there's another one down near the bottom that had a low percentage. Okay, so I'm just going to go over the questions that have a low percentage. Chapter 3 is on cells, so it did contain a lot of information. But anyway, this question says the phosphate head of a phospholipid is blank. So phospholipids have a phosphate head, and then they have a tail of the tail is a fatty acid tail. Sorry, I was trying. It just slipped my mind. But the head is called the phosphate head, and then there are two fatty acid tails that are attached to it. Okay, and remember the phospholipids form like a double layer of phospholipids, a lipid bilayer, and that's what the main backbone of the plasma membrane is. So plasma membrane is composed of phospholipids. The plasma membrane of all cells is composed of phospholipids. So the phosphate head is hydrophilic. In other words, it is soluble in water. It is water loving. And the heads of the phospholipids face outside the cell, where the environment is usually watery, and inside the cell, where the cytoplasm, the fluid inside the cell, is a water-based fluid. So we say that the heads are hydrophilic because they're soluble in water or water-loving. And they are polar because polar covalent molecules are soluble in water. So anytime something is hydrophilic and water soluble, it's always a polar molecule or else an ionic compound. So it's something chemically that can dissolve in water. So that's the answer to this one. Let's look at the other question that I saw. Sorry. Let's get out of that. Here we go. All right, so the other one that I saw kind of a low percentage on, seemed like there was one lower than 50, but I guess not. So let's look at this one that has a 50% by it. And this question says, this is not the question I was thinking of, but this is fine. Which of the following is true of the sodium-potassium exchange pump? So the sodium-potassium pump is a membrane protein. In other words, it's a carrier protein that's found in the plasma membrane. And what it does, it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell at the same time. That's why we call it an exchange pump. This is an example of counter transport. So anyway, it says which is true. This is an example of active transport. That is a true statement because the sodium is pumped out against its concentration gradient. Its concentration is already high outside the cell. So it's continually pumped to where its concentration is high. We learned that for diffusion to occur, molecules are going to move from where their concentration is high to where their concentration is low. But in active transport, molecules are moved toward their high concentration. So sodium is pumped out and potassium is pumped in where their concentrations are already high. So that's an example of active transport, and it also requires ATP energy. Okay, but there's another one that I saw that I want to look at. It's also about transport. Here it is. I didn't see that 25 there. Okay, So how does facilitated diffusion differ from simple diffusion? Well, it has to do with which portion or which area of the plasma membrane that the substance that is diffusing travels through. So if it comes through the phospholipid bilayer, it's simple diffusion. Simple diffusion does not require a carrier protein. But in facilitated diffusion, a carrier protein is required, but it's still passive transport. It doesn't require ATP, and the molecules move toward their low concentration. So facilitated diffusion is different from simple diffusion because it requires a carrier protein. And that's why this answer, which may have confused you a little bit, says the rate of solute transport cannot increase indefinitely in facilitated diffusion. So it cannot it can't diffuse faster and faster at a faster and faster rate because it is limited by the number of carrier proteins. So let's just say, for example, that the solute is glucose. The only way glucose can move into the cell or move, into or out of the cell. We typically talk about glucose moving into the cell, though, because our cells need glucose to carry out cellular respiration and produce ATP. But the rate of glucose diffusion or into the cell is dependent on the number of carriers, the number of transport or carrier proteins for glucose. So if there's 100, then 100 glucose molecules can move in at one time. But if there's only 10 carriers, then only 10 can move in. So it's the rate cannot increase indefinitely because the rate of transport is limited by the number of carriers. And down here, that's why this statement is false. This statement says facilitated diffusion is independent of carrier molecules. For that to be true, it should say facilitated diffusion is dependent on the number of carrier molecules for that substance. So I hope this helped. Let's look at next week, which is week 3. And let's see, that's chapters 4 and 5. We're getting away from review and going back into anatomy and physiology topics. So chapter 4 is on histology, or the study of tissues. The graded assignments are going to be the chapter 4 dynamic study modules, the chapter 4 homework, the lab 4, and then chapter 4 homework quiz. Okay. Now you also notice you have a lecture exam on chapters 1, 2, and 3 that is also due. It will have 50 questions and you will have a time limit of 60 minutes. Then we move on to chapter 5, which is on the skin and associated structures like hair and nails and sweat glands and oil glands. But the skin is part of the main component of the integumentary system. So you're going to do the chapter 5 dynamic study modules for a grade, chapter 5 homework, lab 5, and chapter 5 homework quiz. So all of the chapter 4 and 5 assignments in mastering, and then also lecture exam 1. All right, so I hope you guys have a great week. If you need anything, please email. And also don't forget about the open forum. You guys did it whenever it was a graded assignment. Now that it's not graded, you're not really making use of it. But if it's a question about the material that you're studying for that week, it's really good to post it in the discussion forum or the weekly forum because it helps other students as well as you. All right, so you guys have a great week, and just let me know if you have any problems that I can help you with. Bye.